Hi, I'm Professor Emad,
And I'm Professor Philip.
and welcome to Emad and Philip's Biology Lab World. So, today we were testing organic molecules, specifically carbohydrates, lipids, and proteins. We aren't testing nucleic acids today. The organic molecules that we're testing today are those that you cannot get by your body just producing them. Nucleic acids are the one organic molecule that your body does not need to get anything from a diet. Proteins, for example, your body does produce, but you do need amino acids that your body does not produce. So you need to consume those.
Essential amino acids.
Essential amino acids. So this is a test tube containing Bio-Ret reagent with water. Okay, just distilled plain water and Bio-Ret reagent. There is 20 milliliters of test substance. Okay, that's just the water, and 10 drops of the Bio-Ret reagent. Okay. That was our test subject, and you notice the color. Here we have 20 milliliters of egg albumin, which is a protein, and 10 drops of Bio-Ret reagent. Notice the color showing that the reagent reacted with the protein from the egg albumin. That shows a positive test result. Here we have unknown substance number 1, 20 milliliters with 10 drops of Bio-Ret reagent. Notice the color? That's Bio-Ret reagent in the background Professor Philip is holding. Okay, that's the color of the reagent. Notice how this is not the same color as the egg albumin. From this we can see, because we know this is containing proteins, we know that we see that the colors are different. We know that this unknown substance 1 does not contain proteins. The Bio-Ret reagent reacts with proteins. Here we have unknown substance number 2, 20 milliliters with 10 drops. Notice the color. Similar, correct? This is just a bit more diluted. This is straight protein, so that means this is going to react heavily with Bio-Ret reagent. So we can tell that this is a positive test result, but not as strong as this. This is unknown substance number 3, okay, as compared to egg albumin. Here we have a bit of blue, okay, like this. We kind of diluted it. So we can tell that this reacted well with the Bio-Ret reagents. The positive test result contains proteins. Now notice the color of this. This is unknown substance number 4. Okay, 20 drops or 20 milliliters with 10 drops of Bio-Ret reagent. Notice the color compared to the Bio-Ret reagent. Professor Philip, what is your input? Do you think that this reacted positively? That this contains proteins?
Yes, because one is a murky grape color, while the other is a clear grape color, but they're still both grape colors. So it means that there are proteins in this unknown.
The colors are the same, meaning the Bio-Ret reagent reacted with it. Welcome back,
This is
Philip.
Animai.
Biolab World. Welcome to the Sudan Regents. Not Sudan region. Sudan Regents test for lipids. Here we have the positive test result going with the vegetable oil and Sudan Regent solution. Here we have,
Negative.
this is a negative test result with water. So we can tell this is positive, negative. Okay. Going with unknown substance number 1. Okay. You can see. Looks like the water. Philip, do you mind? Like the water.
Very similar.
Very similar. Okay, we don't see any levels of fat. Don't worry about it. Mr. Philip,
Yes?
do you think that maybe
Where?
that would have some level of fat?
No.
No?
Oh, I'm stuck in the water.
Unknown substance number 2. Compared with this, you can see that it's obviously not a strong factor containing lipids.
You mean that thing?
Really hard to tell though. Okay. But we think, from what we see, unknown substance number 2, no lipids.
No.
Unknown substance number 3.
At first sight, you might say, oh, it's negative because there's no separation.
No separation.
Well,
Apparently.
take a look at this.
Maybe hard to see.
Oh, you can see it.
You can see it.
Right there. Do you see the red?
Unknown substance number 4. Compared to this, first sight, not a lipid.
Oh, actually, it's fructose.
Really hard to see, but there are some micelles there. Globules of fat.
Fructose is fruit sugar.
Something you'll have to notice and to look very carefully for when you're doing the experiment. Water involves many complicated materials, such as ice and water. Ice water. The reaction is more of a feel. Okay, I can feel that the water is getting colder.
No, this is ethanol.
Ethanol and
And
alcohol.
we replace water in ethanol.
Ice in ethanol.
Alcohol.
Alright, I'm ready.
Whiskey on the rocks. Okay. Now you can see that the bubbles coming off. Notice how the ice sinks to the bottom of the ethanol, denser than ethanol. The water, however, the ice floats.
Water has a density of 1.001.
1.00. Density you measure, or in a certain unit you measure compared to water. Specific density. Now, the reason that the ice floats in the water is the ice is less dense. At a certain point, a certain temperature, water becomes solid, and the crystals, the molecules form crystals, crystalline structures, that actually make it less dense than liquid water. However, compared to the ethanol, it's denser.
Which means it would sink down.
Sink. So, if I had an ocean full of alcohol, ice wouldn't stay out.
You can take it out.